So I'm Lars Engstrand. I'm a PI for a research group in Stockholm, Sweden at the Karolinska Institutet. I have an MD background and I have been working with the microbiome since 15 years back. I'm now the director for the Center for Translational Microbiome Research. Well, it's obvious that the microbiome is a dynamic organ, so to say, so it's even within a patient or an individual there is a huge variation, and there are many microbiomes, not only one, from the skin, from the stomach, from the oral tract and so forth. But the dynamic process in one human is quite dramatic, so you have to take that into consideration when you study the microbiome on an individual level. If you move then up to the cohort and the population level, it's a different story, because it's hard to follow the individuals over time, so you have to take one snapshot from that individual. And when you interpret the data, you need to take that into consideration. But there is a huge number of microbes in these organs, and it's a tricky story to really understand how they crosstalk to the human being. It's obvious that we have variations in the microbiome from different populations. We have mostly studied the Scandinavian populations in our center, so the important thing is to get as much metadata or as much information as possible from that population. What kind of living conditions, what kind of dietary factors, and also if you have any drug intake. So we can see that that will have a major impact on the composition of the microbiome. Well, we launched this program in October 2019. Well, we have reached quite many samples now in Europe, but also in China. So I think we reached almost 500,000. But we need to add more, so to say, niches in the human body, not only the gastro. We also want to have the oral tract and the vaginal tract and the skin. So we are aiming for expanding the sites where we sample. Well, the capacity of the T7, for example, can make it possible to really scale up. And we can now see that the different steps in the pipeline is depending on the sequencing capacity, but also the robotic automation is important in the prior steps to the sequencing. So far, the MGI pipeline has fulfilled all the criteria. We are very satisfied with the technology, of course. We have used this since 2017 or 18 in my lab. We have also sequenced more than 50,000 shotgun metagenomes. Everything that is now added to the pipeline will hopefully help us in the microbiome field. And the introduction of this long read capacity will also help us in certain parts of the microbiome research we are performing. To combine short reads with long reads in sequencing of specific bacterial strains will be very, very helpful for single-cell sequencing and spatial transcriptomics. So I can only congratulate the technology.